All right, guys. I'm going to go through meiosis with you a little bit. We're going to go through this whole process that we did in class. I'm not going to redraw all the pictures. Reason being is it took us like, 30, 35 minutes in class, and I don't want to make this video that long. I want to give you guys just what you need to know, and then move on. You can watch the meiosis video. You can watch the mitosis video. We did that whole diagram that compared mitosis and meiosis. You can look at that. We'll go over that a little bit in this video, but I'm going to try to make this as short and sweet as I can. For meiosis, again, guys, we talked about in class, this is only dealing with our sex cells. This is only dealing with sperm in males and eggs in females, okay? The goal of meiosis is to make a haploid cell. For us, 23 chromosomes. That way we can have the sperm, which is 23 chromosomes. We can have the egg, which is 23 chromosomes. When they come together, we got fertilization, 23 plus 23, that's 46 chromosomes, which makes you, your first cell. Just like mitosis, we got the same phases. We got prophase, we got metaphase, we got anaphase, we got telophase. It's the same stuff that happens. Only difference is meiosis, we get two divisions. Mitosis is just one. Meiosis is two. Since there's two divisions for meiosis, we call meiosis the first cell division meiosis 1, and we call the second cell division meiosis 2. Kind of makes sense. Also, the phases, since we had in mitosis prophase, metaphase, anaphase, telophase, which they already had their names, we had to call them something slightly different for meiosis 1 and meiosis 2. So what do? Well, heck, this is called meiosis 1, let's call it prophase 1. This is called meiosis 2, let's call it prophase 2. Makes sense, right? So let's talk about the major events that happen in here. I'm going to show you guys some chromosomes that I kind of made. They're right here. But I'll show you some chromosomes. I'll show you what's happening, especially in prophase 1, because that's probably the most confusing part. And then after that, it gets a little bit easier. So I'm going to start off, and I got 4 chromosomes up here, but I'm going to start off with just showing you one chromosome. Okay? So here's our one chromosome. I got a blue, and I got like a neon yellowish green color. You guys already know that after a cell is done dividing, it's going to go through interphase. That same interphase, the G1, the S, the G2, those are our 3 stages of interphase, they happen again for meiosis. So before the cell goes through meiosis, the chromosome or the cell is going to go through interphase. So during G1, it's going to grow. During S phase, the DNA is going to replicate. Now, you know during S phase, it's chromatin. So it's long, stringy. I can't show you that, okay? It'd be ridiculously long, and I just don't feel like making a piece of string that long. So I'm going to show you the chromosome. So here's our chromosome for this cell, all right? What we're going to do is during S phase, we're going to make a copy of it. And again, guys, right now it's chromatin, all right? So now we have our copy, all right? I guess inverted, so there you go. So we have our copy, not exactly alike, different little different shapes, but same genes, same traits. We're going to have that for prophase here, okay? Now, what's going to happen for prophase is the chromatin is going to turn into chromosomes, these guys. So we got a copy of each one. And the big thing you need to know for prophase 1, the biggest thing for all of meiosis that's really different than mitosis is crossing over. Here's what happens for crossing over. We're going to take these 2 chromosomes, and we're just going to literally switch some traits up, okay? When we're talking about these chromosomes, guys, let's say you got this one from your mom, and you got this one from your dad. Your mom's DNA and your dad's DNA, we're going to essentially change them up a little bit. So for these 2 here, let's say that crossing over occurs, okay? So we're going to switch some traits up. What's going to happen is it's no longer going to look like this. Instead, we're going to have something like this, all right? You guys can see that down here, some of the mom's chromosome or chromatid went to the dad, and then some of the dads went to the mom. They flip-flopped. And on the other one, again, we replicated it. We're also going to have crossing over. Sorry that my hands are in the way, guys. That's a little better. We're also going to have crossing over, but maybe it happens at a different location. So now we have 4 completely different chromatids. And that's the big thing with prophase, guys. You have to remember that crossing over occurs, and that's it. All right? That's the only time it happens for all of meiosis. It's the only time crossing over happens. Also, the nuclear membrane disappears. That's it. All right? After that, let's go through Metaphase One. What's going to happen here, guys, is we're going to have tetrads. All the tetrad is again, here's our chromosomes after crossing over. They're going to line up in the middle, just like that. So they're right there lining up in the middle. And that's it, all right? Most ones are going to have more than one chromosome. I know I have just one here, and we copied it, so we have 2, but most organisms are going to have more than one, but for this part, we're just going to pretend it's one, just makes it simpler on us. And after that, after metaphase, all right, they're lined up in the middle, we're going to have these centrioles and the spindle fibers attached, and what they're going to do is they are going to take these chromosomes and pull them apart to opposite ends of the cell. And that's what happens in our anaphase there. All right, they just pull apart different parts of the cell. Telophase, if this is an animal cell, we're going to have our cleavage furrow again. If it's a plant cell, it's the cell plate, but they're going to just make the 2 new cells, the nucleus reforms, and that's the end of meiosis 1. That's all that happens. So at the end of meiosis 1, we have 2 cells, okay? We have 2 cells, and right now, their chromosome number is still essentially 46 chromosomes per cell, all right? Because we had DNA replication happen at the beginning here, so we have 46. What we're going to do is we're going to take this exact this exact 2 cells from telophase 1, and we're going to take them over and put them right into meiosis 2. Interphase does not occur again between meiosis 1 and meiosis 2. We literally just transport these right into the next prophase, all right? So during prophase 2, almost the same thing happens as in regular prophase. The nuclear membrane disappears. Only difference here, guys, is that crossing over, remember the traits switching up like they did in prophase 1, does not occur. So again, during prophase 2, crossing over does not occur. Crossing over only happens once, and that's in prophase 1. I think I've mentioned that like 8 times already, crossing over. Pretty important, right? After we have this, okay, we're now going to have, these are going to be on opposite cells, but we're going to have metaphase. They're lining up in the middle once again. All right, nothing new there. We just got 2 cells this time. They're a tetrad up here for metaphase 1, but for metaphase 2, they're just chromosomes, all right, individual chromosomes. This is going to allow these to separate during anaphase 2. So during anaphase 2, guys, of meiosis 2, what happens is our chromatids, TID, separate. So we had a chromosome, now we got chromatids, and that's all for anaphase 2. Lastly, telophase 2, we're going to form our 4 cells, right? For spermatogenesis, all these cells are going to be the exact same size. That's in males. For oogenesis or oogenesis, however you want to say it, in females, they are going to be different sizes. We're going to have 1 large egg, and the other 3 are called polar bodies. They're much smaller. They will not be used for eggs, all right? Now, if you look at my 4 chromatids that I have here, you can see that this yellow one, this would be in 1 cell, is much different than this yellow one because the crossing over exchange different traits. They're at different locations. This is completely different traits for you guys. So this one might be here, this one might be here, and then our other 2 blue ones, which are still different than one another, one might be here and one might be here. Why I'm showing you that, guys, is that all 4 of these are different. This means that all of our cells at the end of telophase are completely different. And that's the goal of meiosis, to produce 4 genetically different cells. This is so you don't look like your parents, you don't look like your grandparents, you don't look like your brother or your sister. Every single time fertilization happens after this, between the sperm and the egg, we are going to have 4 cells that are completely different. In other words, they have a different DNA makeup. And that's why meiosis is so important, because we still get genetic variation due to our crossing over. So that way, again, we don't look like our parents. If you remember, mitosis produced genetically identical cells. We wanted them to be identical because we want our heart cells to stay heart cells. We don't want them to be different. We don't want them to change into a lung cell or what have you, okay? We want that cell to always be a heart cell because it has a specific job to do, all right? For these ones, the goal of the sperm cells and egg cells is simply to make a new human so that we have the two haploid cells, haploid from the sperm, haploid from the egg, coming together for fertilization to make that diploid cell, all right? At the end of meiosis 2, guys, these are haploid cells. For us, they have 23 chromosomes. That's different from mitosis because mitosis had 46 chromosomes and those were diploid. Another way to say this is these are N, okay? N is like first letter in number. What that means is they have one copy of each chromosome. For diploid or mitosis, we call that 2N, which means that they have two copies of each chromosome. You guys remember we have 23 chromosomes, just like that website, 23andme.com. That tells us that we have 23 different chromosomes. Mitosis, you have 2 of each of those 23. Meiosis, you only have 1 of them, all right? So again, big points for this, guys. Make sure you know crossing over is during prophase 1, all right? Make sure you know we do 2 meiotic divisions, all right? We do 1 for meiosis 1, the second division for meiosis 2. Overall, we get 4 cells in the end, all right? Different than mitosis because mitosis, we only get 2 cells in the end, and those cells are diploid, all right? Hopefully this helped a little bit, guys. You can watch this video along with the mitosis video. You should be good for tomorrow.